My name is Dan Johnston, and I'm director of the Institute for Neuroscience, as well as the director of the Center for Learning and Memory. I'll explain each as I go along. The Institute for Neuroscience is the intellectual and administrative home for neuroscience research and neuroscience graduate education across campus. We have well over 70 faculty members who are affiliated with the Institute for Neuroscience. I think there are 7 different colleges represented within the Institute and quite a number of departments. The purpose of the Institute is to foster neuroscience as an independent discipline on campus. We sponsor graduate courses, graduate education. We run a PhD program in neuroscience out of the Institute. We have symposia and lecture series, and we try to foster collaborative projects and research. The Department of Chemistry has one faculty member, Jason Scheer, who's a member of the Institute for Neuroscience. But there are a large number of chemistry faculty who collaborate with Institute Neuroscience faculty members, including Dio Siegel in chemistry collaborates with Kimberly Rapp-Graham. Steve Martin collaborates with Richard Aldrich. And we hope to see these collaborations increase in the future as neuroscience will move into the new Norman Hackerman building, as well as a portion of chemistry. So we'll be ultimately in the same building, and we hope to continue fostering collaborations and leading to new discoveries related to the chemistry of the brain. Now I'm also director of the Center for Learning and Memory. This was established about 5 years ago by the provost and has been the means for major growth of faculty on campus in the area of neuroscience. So we were given at least 10 positions to hire new faculty members, and these faculty members could be housed in any department that would be most appropriate for their discipline. And the logic behind the Center for Learning and Memory was learning and memory is a central feature of the brain, and it encompasses molecular studies through behavior. And so the goal was to hire faculty members who would span these different research areas and foster collaborations with existing faculty members on campus. Ones, for example, more molecularly oriented would collaborate with biochemists and molecular biologists and geneticists on campus. Cellular and systems-oriented research people would collaborate with people in psychology and neurobiology, and also the systems people would collaborate with potentially people in engineering, biomedical engineering, electrical engineering, as well as computer science and so forth. So far it's been very successful. We've hired 8 faculty members, and almost every one of them has formed collaborations with one or more of these groups, and we hope to continue our recruitment efforts. We probably have 4 positions yet to fill, and we'll be continuing to recruit over the next few years. And one component will also be with an eye towards translational research, basic research that can be most easily and quickly translated into potential therapies and cures and better understandings of some of the devastating diseases of the nervous system, both diseases of development, diseases of aging, and in particular diseases that affect learning, memory, and cognitive abilities of the individual. and many of our faculty members have projects that relate to this, and we hope that we can collaborate with Texas Institute of TI3D in using some of our basic research discoveries to develop drugs that would target particular molecular and systems entities in the brain.
I'm Jason Shear. I'm a professor of chemistry, and my lab in Welch is involved in a range of different projects, some of which really touch on the mission of the Institute of Neuroscience. An area that we've had long-standing interest in is trying to understand the functioning of nerve cells at the level of small numbers of cells. This extends from trying to understand how the cells communicate with each other to how they grow and develop. And one of the approaches that we've taken is to try to develop culture environments, so isolated environments away from the in vivo systems that will allow us to study cells under more controlled conditions. But we've also wanted to try to maintain more of the relevance to how cells function in the body that culture environments typically allow. So my group's been developing a range of different biomaterials for culturing cells in and alongside that will serve as three-dimensional scaffolds, three-dimensional environments for looking at cell growth, migration, and interaction with other nerve cells.
Hi, I'm Stephen Martin. I'm the Wagner Chair of Chemistry and Biochemistry, and one of the exciting projects I'm involved in is working with Rick Aldrich in the Institute for Neuroscience. He has identified a small molecule, an alkaloid in fact, that was found in a marine organism that modulates the opening and closing of potassium channels. And so one of the things that we would like to do in this collaboration that we've established is prepare molecules, small molecules that are derivatives of this natural product, but which are more stable in solution, so that we could eventually obtain crystal structures of complexes of this small molecule with potassium channels themselves. And if we can do that, then perhaps we can get some insights on how small molecules modulate the opening and closing of these channels.